Hello. So now we're going to see about apoptosis or apoptosis, better pronounced. So talking about apoptosis, so in this image we can see leaves are falling, right? So apoptosis is derived from the Greek word which means to fall off. We can see here the leaves they are falling away. So what happens in this thing is when the leaf in a tree it grows old, is no longer able to sustain himself. What will happen? The leaf will just fall down. See, the leaves will just fall down. These leaves, they've done their function. They've achieved their life goals. Now they are falling away. So they just fall off. Ptosis is to fall off. Apo is self. Thus they will fall off on the self without an external thing. Leaves are done. They just fall off. So talking about apoptosis, we have two pathway. One is intrinsic pathway, one is extrinsic pathway. On intrinsic pathway, it is based on mitochondria, completely based on mitochondria. It starts from inside the cell. And extrinsic pathway is based on the external features of the cell. So talking about apoptosis, it's an energy dependent, genetically programmed cell death. It can involve a single cell or a group of cells. Example is endometrial shedding or removal of cells during embryogenesis. Then what happens in apoptosis? That is, the cell will shrink, cytoplasm will become more eosinophilic. These are the appearances. So let's see the mechanism, what actually happens over here. So what I have here is the intrinsic pathway and the extrinsic pathway. So as we are able to appreciate over here, the intrinsic pathway starts from inside the cell. It will happen something will happen, and it will eventually destroy my cell. And the extrinsic pathway, the same thing will happen. That is, the cell will be eventually destroyed, but it starts from outside the cell. So what happens in intrinsic pathway? There are growth factor can be withdrawal. There can be DNA damage by radiation, toxin, free radicals like nitrogen or anything for that oxide species, or misfolding proteins. So there are in the intrinsic phase there are anti-apoptic, pro-apoptic, and sensory cell proteins are present. So we shall be going over them shortly. And talking about death receptors, that is the extrinsic pathway, I have FS and TNF receptors, which will start the initiator caspase and eventually stimulate the executioner caspase. That is the caspase 8 and 10. They are the executioner caspase. They'll finally execution the task and they will execution the cell. Execute the cell. That is the death of the cell shall be executed by them. So talking about the intrinsic pathway, I have anti-apoptic genes like BCL2, BCL, MCL, pro-apoptic, that is back and BCLX3, and sensory cell proteins like BIM, BID, and BAD. So what happens at the surface of the cell site? Normal, we'll see what happens in normal cell first. In normal viable cell, I have a growth factors that is there. It will continue production of an anti-apoptic protein. See, for example, this over here, I can see the BCL2, for example, in this case. This BCL2, what will happen? This BCL2 will give a signal key, don't let the cytochrome C out, leak out. See over here, this is the BCL2. What will happen is this BCL2 in the cytoplasm will prevent leaking of the cytochrome C which is inside the cell. See, there is no leakage of cytochrome C. So what happens? Say the surviving factor, that is the growth factor or any factor that was for that causing the cell to remain, it is removed, or I give radiation or something else. So what will happen? The BH3 proteins, that is the sensory proteins, that is as I told earlier, BIM, BID, and BAD. What happens? These will be stimulated. They will sense that BCL2 is gone. As BCL2 will be gone, we can see the cytochrome C will just move out of the cell. As there is leakage of cytochrome C, what will happen? The pro-apoptic genes, that is backs and back. You can see here the backs and back, which are the pro-apoptotic genes. They will be activated. So cytochrome C and other proteins. So you can see in the endoplasmic reticulum, if there is a just like a tunnel or some opening is there, all the contents from the inside they will just leak and go outside. This will cause activation of caspase. So what are caspase? Talking about caspases, they are proteins. They are proteins which are activated by a chain reaction and they eventually cause the cell death. So this is what happens in the intrinsic phase. That is, if there is some DNA error which can stimulate the BIM bad or the BID proteins, which will cause two things. One, it will destroy the BCL2, that is the antagonism of BCL2. And secondly, it will activate the Bax channel. So the first thing, it will cause the destruction of BCL2. Second, it will activate the Bax channel. So due to this, cytochrome C will be leaking out. As cytochrome C will be leaking out, the cytochrome C will go out of cell and this shall stimulate the caspase, that is the caspase 9, activation of caspase 9. Caspase 9. So they will go and stimulate the execution of caspase. This is what happens in the intrinsic mechanism. So next, talking about the extrinsic mechanism. This is the extrinsic mechanism. What happens over here? It will start from outside. Say, for example, a virus or something came and it will attach to the FAS. FAS is a receptor present in the cell. So FASL, that is the FAS ligand, it will come and attach to FAS. FAS or CD95, it belongs to the TNF superfamily. So once FAS and FAL attaches, they will activate the FADD, that is fast associated death domain. Once the fast associated death domain is stimulated, they will stimulate procaspase 8 or 9. This will again cause active formation of caspase 8 or 9. This will stimulate the execution of caspase, which will cause apoptosis. So this is what happens. So talking about it, FADD, that is fast associated death domain. Death domain, these are death domains. They cause leaking out of the contents, which will eventually cause the death. That is the death domain. So I would like to take a note and mention about necroptosis. That is the necroptosis. It is a mixture of necrosis and apoptosis. So it is a programmed cell death. We have kinases which like RIPK1 and RIPK3, which will cause this. What happens after this? Once the TNF R1 will recruit this kinase in a multiprotein complex of various proteins shall be formed, which will be made of this RIPK. And over there, once this TN TNFR1 comes and binds over here, that is a TNFR receptor will be present. Once TNF comes and binds to the receptors, what will happen is MLKL is a protein that is present. MLKL protein will start polymerizing. Once this start to polymerize, it will cause the breakdown of the membrane. So what it will do, over here we will have the membrane. It will come and it will just remove the membrane. It will just break the membrane, and that is a very characteristic feature of necrosis. That okay, we remove the plasma membrane, right? That is a very characteristic feature of necrosis. So next, talking about apoptosis, we have something called as apoptic body. So apoptic body is single cell. It has dark eosinophilic nuclei. So morphologically, it is a shrinking cell which lead the cell to become more eosinophilic. Nucleus condensed in an organized manner, and due to which apoptic bodies fall down from the cell, and later this apoptic body. So over here we can see a apoptic body, intensely eosinophilic. The nucleus size has been reduced. This is on the left hand side of the A. It is a H&D stain. We can see apoptosis. This is apoptosis of epidermal cell in the immune reaction, and it has a brightly eosinophilic cytoplasm and very condensed nuclei. Over here we have a normal cell. We are able to approach normal cell. We can see that the color, and we can see the color of this. On the RH, the right hand side, I have a cultured cells and I have electron microscopic images. So this is the nucleus, the dark color one. I'm able to observe crescents on the electron microscopy image. This is along with compacted chromatin. These are uniformly dense like this, or they are fragmented. Next, I shall be talking about another image, that is IHC, immunohistochemistry. So over here I have on my left hand side, that is on the first panel, I can see blebbing. Blebbing is a change in the surface that is small. It is the first reversible earlier signs that is blebbing. We can appreciate blebbing over here. On in the middle panel, I can see phase contrast micrograph, and I can show the DNA fragmentation over here. The DNA gets fragmentation, and this is the IHC, immune histochemistry. I have a immunofluorescence stain with a red color dye. This red color dye, I have it with the red color dye. This will help me to identify it. So over here I have the red color IHC for Caspase 3. Just highlighted, right? So over here for Caspase 3, this is giving me the characteristic red color that I can see over here. So yes, with this I'm going to end it. Thank you.